Another day, another story. The discovery of DNA is a complex and multifaceted story that spans several decades and involves the contributions of various scientists. While the structure of DNA was ultimately elucidated in the mid-20th century, the groundwork for this discovery was laid in the 19th century. Here is a brief overview of the key developments leading up to the understanding of DNA. Welcome to Tabo Eminent Channel. Isolation of Nuclein, 1869. The journey begins with Swiss physician and biochemist Friedrich Miescher. In 1869, Miescher isolated a substance from the nuclei of white blood cells, which he called nuclein. This discovery laid the foundation for the identification of genetic material. Chromosomes as carriers of genetic material, 1888 to 1905. In the late 19th and early 20th centuries, researchers began to associate hereditary information with the structures known as chromosomes. Walter Fleming observed the process of mitosis and described the presence of thread-like structures that would later be identified as chromosomes. Identification of nucleic acids, 1909 to 1928. Phoebus Levene made significant contributions to the understanding of nucleic acids during this period. He identified the basic components of nucleic acids, such as nucleotides and the sugar phosphate backbone. Levene's work helped establish that nucleic acids were composed of repeating units. Transformation principle, 1928. Frederick Griffith's experiments with bacteria in 1928 provided evidence of a transforming principle. Griffith observed that a non-virulent strain of bacteria could be transformed into a virulent one when in contact with the heat-killed remains of a virulent strain. This hinted at the transfer of genetic material. Avery, MacLeod, and McCarty, 1944. Building on Griffith's work, Oswald Avery, Colin MacLeod, and Macklin McCarty conducted experiments that identified DNA as the substance responsible for transformation. They demonstrated that when they destroyed the DNA in a virulent strain, the transforming ability was lost. Chargaff's rules, 1949. Owen Chargaff's research revealed consistent ratios of adenine to thymine and cytosine to guanine in DNA samples. This observation, known as Chargaff's rules, hinted at the specific base pairings within the DNA molecule. X-ray crystallography, 1950s. Rosalind Franklin's work using X-ray crystallography provided crucial insights into the structure of DNA. Her photographs, including the famous Photo 51, were instrumental in deciphering the helical structure. Watson and Crick's model, 1953. In 1953, James Watson and Francis Crick, with the help of Rosalind Franklin's data, proposed the double helix structure of DNA. Their groundbreaking model described how the two strands of DNA are held together by complementary base pairs, adenine with thymine, and guanine with cytosine. The discovery of DNA was a collaborative effort, with many scientists contributing to different aspects of the puzzle. Watson, Crick, and Wilkins were awarded the Nobel Prize in Physiology or Medicine in 1962 for their groundbreaking work in deciphering the structure of DNA. Rosalind Franklin's contribution, 1950s. Rosalind Franklin's X-ray crystallography images, particularly Photo 51, played a crucial role in understanding the structure of DNA. Despite not being directly involved in the model building process with Watson and Crick, her work provided vital information about the helical nature of DNA and the distance between the phosphate backbones. Watson and Crick's model, 1953. James Watson and Francis Crick, based at the Cavendish Laboratory in Cambridge, used Franklin's data along with Chargaff's rules to construct their groundbreaking model of DNA. On February 28, 1953, they proposed the double helix structure in a short paper published in the journal Nature. Their model elegantly explained how genetic information could be stored and replicated. Publication of the DNA structure, 1953. The publication of Watson and Crick's model marked a turning point in biology. The structure of DNA provided a clear understanding of how genetic information is passed from one generation to the next. Their paper, along with a second paper by Maurice Wilkins and Rosalind Franklin's colleague Raymond Gosling, was published back-to-back in the same issue of Nature. Impact and Recognition, 1962. In 1962, James Watson, Francis Crick, and Maurice Wilkins were awarded the Nobel Prize in Physiology or Medicine for their discoveries related to the molecular structure of DNA. Unfortunately, Rosalind Franklin had passed away in 1958 and, according to Nobel Prize rules, the award is not given posthumously. Further Understanding and DNA Replication, 1950s-1960s. Following the elucidation of the DNA structure, scientists turned their attention to understanding the mechanisms of DNA replication and the role of DNA in heredity. The work of Arthur Kornberg and his team in the late 1950s, for instance, demonstrated the enzymatic synthesis of DNA. Expansion of genetic code knowledge, 1960s. The 1960s saw further exploration of the genetic code. Marshall Nirenberg and Hargobin Khorana deciphered the genetic code, revealing how sequences of nucleotides specify the amino acid sequences in proteins. Discovery of DNA polymerase, 1970s. The discovery of DNA polymerase by Kornberg and Thomas A. Baker in the early 1970s was a significant step in understanding the process of DNA replication. DNA polymerase is an enzyme responsible for synthesizing new DNA strands during replication. The discovery of DNA opened up new avenues for research, leading to advancements in molecular biology, genetics, and medicine. Understanding the structure and function of DNA laid the foundation for the biotechnological revolution and various medical applications in the subsequent decades. The story of DNA discovery is a testament to the collaborative efforts of scientists across different disciplines. RNA's role, 1960s-1970s. Scientists, including Sidney Brenner, François Jacob, and Jacques Monod, expanded the understanding of genetic regulation by investigating the role of RNA in protein synthesis. They discovered messenger RNA, mRNA, transfer RNA, tRNA, and ribosomal RNA, RNA, elucidating the complex process of gene expression. Recombinant DNA technology, 1970s. The development of recombinant DNA technology in the 1970s allowed scientists to manipulate and engineer DNA. Paul Berg, Herbert Boyer, and Stanley Cohen were instrumental in the creation of the first recombinant DNA molecules, paving the way for genetic engineering and biotechnology. DNA sequencing, 1970s-1980s. Friedrich Sanger and his colleagues developed methods for DNA sequencing in the 1970s. These groundbreaking techniques allowed scientists to determine the precise order of nucleotides in a DNA molecule, facilitating the mapping of entire genomes. Human Genome Project, 1990s-2003. The Human Genome Project, initiated in 1990, was a collaborative international effort to sequence and map the entire human genome. This ambitious project involved numerous scientists and institutions and was completed in 2003, providing a comprehensive understanding of the human genetic code. CRISPR-Cas9 gene editing, 2010s. The discovery of the CRISPR-Cas9 gene editing system in the early 2010s revolutionized genetic engineering. Jennifer Doudna and Emmanuelle Charpentier, who received the Nobel Prize in Chemistry in 2020, played key roles in developing this technology, enabling precise and efficient editing of DNA. Functional genomics and systems biology. Advances in functional genomics and systems biology have allowed scientists to study the functions of genes in the context of entire biological systems. This holistic approach aims to understand how genes interact and function together to regulate cellular processes. Epigenetics, 2000s present. The field of epigenetics explores changes in gene expression that do not involve alterations to the underlying DNA sequence. Researchers have uncovered the importance of epigenetic modifications, such as DNA methylation and histone modifications, in regulating gene activity and cellular functions. Precision medicine, 2010s present. The integration of genomic information into medical practice has given rise to the concept of precision medicine. Understanding an individual's genetic makeup allows for personalized diagnosis, treatment, and prevention strategies based on their unique genetic profile. The discovery of DNA has evolved into a rich and dynamic field that continues to shape scientific inquiry and medical advancements. From the elucidation of the double helix structure to the development of powerful gene editing technologies, the story of DNA discovery is ongoing, with new chapters being added regularly as scientists uncover the complexities of the genetic code and its implications for human health and biology. Telomeres and telomerase, 1980s-1990s. Elizabeth Blackburn, Carol Greider, and Jack Shostak made significant contributions to our understanding of telomeres and the enzyme telomerase in the 1980s and 1990s. Telomeres are protective caps at the ends of chromosomes, and telomerase plays a crucial role in maintaining their length. This research has implications for aging and cancer. RNA interference, RNAi, 1990s-2000s. Andrew Fire and Craig Mello discovered RNA interference, RNAi, in the late 1990s. This natural process involves the silencing of gene expression by small RNA molecules. RNAi has become a powerful tool in molecular biology and has applications in gene function studies and potential therapeutic interventions. Functional genomics and high-throughput sequencing, 2000s present. Advances in high-throughput sequencing technologies have allowed scientists to sequence DNA at unprecedented speeds and lower costs. This has led to the field of functional genomics, where researchers can study the functions of genes on a large scale. Synthetic biology, 2000s present. Synthetic biology combines principles from biology and engineering to design and construct new biological parts, devices, and systems. Scientists aim to create synthetic organisms or redesign existing ones for practical purposes, such as producing biofuels or developing new medical treatments. Single-cell genomics, 2010s present. Single-cell genomics has emerged as a powerful technique to analyze the genetic material of individual cells, providing insights into cellular diversity and heterogeneity within tissues. This technology has implications for understanding development, disease, and personalized medicine. DNA storage, 2010s present. Researchers have explored the potential of using DNA as a data storage medium. DNA's ability to store vast amounts of information in a compact and durable form has led to experiments where digital data, including books and images, are encoded into DNA sequences. Genomic medicine and personalized therapies, 2010s present. Genomic medicine is transforming healthcare, with an increasing focus on personalized therapies based on an individual's genetic makeup. Targeted therapies and precision medicine are becoming more common, allowing for tailored treatment plans for various medical conditions, including cancer. Ethical and legal considerations. As the field of genomics advances, there is a growing emphasis on addressing ethical, legal, and social implications. Issues related to privacy, consent, and the responsible use of genetic information are actively discussed, emphasizing the need for ethical frameworks in genomics research and applications. The ongoing story of DNA discovery and genomics is dynamic, with continuous advancements shaping our understanding of life at the molecular level and influencing various fields, from medicine to biotechnology. As technology and knowledge continue to evolve, the story of DNA unfolds in new and unexpected ways, promising further breakthroughs and applications in the future. Thanks for watching. Request you to subscribe the channel.